Greeting to Dr. Muhammad Effendi. My name is Roslana binti Rafael. My number matrix 106168. I will present my mock proposal final year 1 project. My title is Effect of Crotalaria juncea cover crop residues on the early growth of Zea mays L in Borneo. For introduction, Zea mays L is an important cereal crop for food, feed, and industry. Excessive use of chemical fertilizers may cause soil degradation and environmental pollution. Legume cover crops such as Crotalaria ginsea can improve soil fertility naturally, which represent a sustainable agricultural practice. Next, the problem statement. Crotalaria ginsea known as a good organic fertilizer. However, during the decomposition phase, these residues may also cause temporary allelopathic effect and nutrient immobilization. This will make farmers may hesitate to adopt residue-based nutrient management. Crotalaria ginsea residue effect on early growth of maize also has a limited studies in Borneo. This knowledge gap is necessary to evaluate the sustainable alternatives in the use of Crotalaria ginsea residue on early growth of Zea mays L. There are three objectives: one, to determine the effect of Crotalaria ginsea cover crop residue on the germination percentage and early growth of Zea mays L; two, to evaluate the effect of Crotalaria ginsea residues on the early growth parameters of Zea mays L, including plant height, leaf number, and biomass; three, to assess the potential of Crotalaria ginsea residues as a sustainable organic nutrient source of maize production in Borneo. There are four literature reviews. 1. Zea mays L and its agricultural importance. In Malaysia, maize is highly priority source to produce wealth for the country, especially formation of animal feed. For human consumption, maize can be used in food pyramid, such as carbohydrate source that is good for human health. Maize also important raw material for industrial application, such as starch, biofuel, and can be side income for farmers. Second, legume cover crop in sustainable agriculture. Legume cover crop can fix atmospheric nitrogen through symbiotic relationships, which can increase soil nutrient availability. These plants can contribute organic matter in soil, improving structure, moisture retention, and microbial activities. The example for legume cover crop is Crotalaria ginsea. Third, Crotalaria ginsea as a cover crop residue. Crotalaria ginsea, also known as sunhame, native to Malaysia, which belong to the family Fabaceae. During decomposition, Crotalaria ginsea residue can contribute organic matter and release essential nutrients which support the growth of maize crops. But the decomposition may also temporarily allelopathic effect which could influence early growth. Lastly, allelopathic effect. Allelopathic can release chemical compounds from plants' residue that may positively or negatively affect the early growth of other plants, especially during residue decomposition. Allelopathic chemical may be released into the soil and influence seed germinations, root development, and nutrient uptake. But when residues are applied without sufficient decomposition time, this effect may result in delayed germination or reduced seedling growth. Material and methods. First, study area. This study will be conducted in University Malaysia Sarawak under controlled greenhouse conditions, which suitable for evaluating this research. The experimental site is located in Borneo and experiences a humid tropical climate. There are four plant materials. First, Crotalaria ginsea residues. Second, 36 maize. Third, topsoil, which collected from agricultural field. Lastly, 12 polythene bags will be used for experimental planting. Experimental design. A completely randomized design (CRD) will be used for data collection because the study will be conducted as a polythene bags under controlled environment conditions, which allowing treatment to be assigned randomly. From the table, there are four different treatment application rates of Crotalaria gentiana, and each treatment will be replicated 3 times, with total of 12 polythene bags used. The preparation and application of Crotalaria gentiana residues. Fresh Crotalaria gentiana will be collected, air-dried, and chopped into small pieces before mixed into the topsoil according to the assigned treatment rates. The amended soil will be left to decompose for 7 to 14 days before planting. After the decomposition period, maize seeds will be sown in polythene bags at one. For planting procedures, first, for Crotalaria gentiana, residues will be chopped into small pieces and left to decompose for 7 to 14 days. The residue then will be mixed to the topsoil according the assigned treatment levels. After that, Zea mays L seeds will be sown in 12 polythene bags that fill with topsoil at 1 cm depth. Next, the 12 polythene bags must be labeled according to their treatment and replicates. Then, watering must be done regularly and observation were recorded throughout. For datasheet to evaluate early growth performance of maize under different treatment levels, the following parameter will be measured. First, the table 3.3 germination percentage of Zea mays Ls under different Crotalaria gentiana residue treatment levels. The germination percentage will be calculated using this formula. The table of 3.4, the plant age within a weeks after sowing the seeds of Zea mays L will be measured. Next, table 3.5, the number of leaves Zea mays L within a week after sowing will be recorded by counting fully developed leaves. Lastly, table 3.6. Fresh and dry biomass of Zea mays L will be measured at the end of the experiment. The last material and method is data analysis. Collected data on growth of Zea mays L will be presented as mean, standard deviation for each Crotalaria gentiana residue treatment. A one-way analysis of variance, ANOVA, will be used to determine whether there are significant differences among the treatment. If significant differences are detected, Tukey Honest significant difference THSD test will be applied for mean comparison of significant levels. A statistical analysis will be performed using R software. For my expected outcomes, the application of Crotalaria gentiana residues is expected to improve the early growth performance of Zea mays L, particularly in germination percentage, plant height, leaf number, and biomass production. Findings from this research are expected to provide useful information on the suitability of Crotalaria gentiana residues as a suitable organic nutrient source for maize production in Borneo. Overall, this study may contribute to environmentally friendly agricultural practices by reducing dependence on chemical fertilizers. This is my work schedule, and this is my reference. Thank you.